Hello students, welcome to Learner's Planet. We are starting next session of subject biology and the topic we are discussing is fundamental unit of life. Till now, in the previous sessions, we have studied the basics of the cell, the fundamental unit, the differences that occurs in the different type of cells, and then we had started with studying the structure of the cell. In that we said all cells are made up of some basic common components. What are they? They are a plasma membrane or a cell membrane, then cytoplasm and the nucleus. The outer boundary of each cell is the plasma membrane. We have studied the entire detail of this plasma membrane, how it behaves, how its flexible nature supports the life of the cell, and whatever point important related to plasma membrane we have studied. Now then we said that plasma membrane is present in all the cells, but in the case of plant cell, this cell membrane or the plasma membrane, it is further covered by the another layer, and that another layer is known as what? That is known as cell wall. This cell wall is a thick covering which is present in surrounding the plant cell. As you can observe that in this particular picture, this inner boundary line, this brown color line, this is what? This is the plasma or the cell membrane, which is enclosing the various organelles, cytoplasm, etc. Outside this another layer is also present, and this another layer is known as what? It is known as cell wall. This cell wall is absolutely absent in the case of animal cells. It is not present in the animal cell, but it is observed in the bacterial cells as well as in the fungal cells also. Although there is some structural and composition variations in these cell walls, in the structure or the chemical composition of these membranes. In different organisms, they are made up of different type of substances. But one thing is very true. It is that wherever the cell wall is present, it is made up of complex polysaccharides, or we can say they are made up of the polysaccharides. In the case of plants, it is made up of cellulose. In the case of fungi, it is made up of chitin. In bacteria, it is made up of a complex polysaccharide, that is peptidoglycan. Now, these chemical composition is present, but definitely they are going to support these particular organism. Whether it is plant, fungi or it is bacteria, they get support because of the presence of such type of extra membrane in its surrounding. See, we have already studied about different type of solutions like hypotonic, hypertonic and isotonic. Hypotonic means what? It is a dilute condition in which the surrounding conditions, they are having more amount of water and less amount of solute as compared to the cell which is placed in that solution. Hypertonic, on the opposite side, it is containing more amount of solute and less amount of water or the solvent. So we have studied that how these hypotonic and hypertonic solution shows actually affects the movement of water molecule. In the case of hypotonic condition, the water is high in the surrounding condition, so it tends to move from outside to the inside, and therefore the cells, they shows endoosmosis and they get swell up. Whereas in the hypertonic, the reverse thing happens. The water which is present inside the cell, it tends to move outside because in the surrounding region the water concentration is lower. So they shows exosmosis, and as a result the cells will get shrink. Now in the case of plant cells, the phenomena takes place. Similar phenomena occurs, but there is certain difference. What the difference is, that is what we are going to study now, and those differences are because of the presence of these polysaccharide cell wall. Let us assume, suppose there is an organism, let us say if it is bacteria, if it is placed in a hypertonic condition, or let us assume it is a plant cell, when it is surrounded by high amount of water, can you think what will happen? Let us understand with the help of this diagram. Suppose this is a plant cell. This is the cell wall, and the inner membrane, this is what? This is a plasma membrane. Inside that, nucleus and other parts would be present. Now, if it is present in the hypertonic medium, that means what? The water is high in surrounding condition. So what will happen? The water molecule, it tends to move inside the cell, inside the vacuole. It will fill up this vacuole inside. So what will happen? The composition or the bulginess, or we can say the extent of this water will increase inside, and that will give some bulginess to the internal structure. And the process, if it continues, then what will happen? The cell will tend to burst. See, in the next diagram we can show, it is, suppose it has gone up to a higher limit. It is swollen up completely. Now what will happen? Water is going inside. Now what happens? Now actually an equal, from inside the water content is increasing, the pressure is increasing, so that pressure will start pushing on this cell wall. And the cell wall, since it is rigid in nature, so what will happen? It is dead in nature, it is rigid in nature, it is non-flexible in nature. So it will exert an equal and opposite pressure. From inside, this pressure is being exerted, and from outside the cell wall will put an equal and opposite pressure against the movement of this particular protoplast. Due to this, what will happen? The cell is not able to burst out further. It will remain intact inside. And this is how the particular cell wall which is present in the surrounding this particular cell will actually help the organism to tolerate the dilute conditions or the hypotonic conditions in surrounding. Normally, if animal cell is placed in such a hypotonic condition, then in that case what will happen? A time will come when that flexibility or the plasma membrane is going to burst out. It is flexible, it will keep on stretching, but a time will come when they cannot further tolerate more stretchness over there, and thereby it is going to burst. And of course, the inner cellular material will come out and the cell will die. So such type of bursting is not observed in the case of plant cell. Why it is so? The reason is due to the presence of the cell wall. So this is how it supports the life of these unicellular organisms, that is fungi and bacteria, as well as plants which are exposed to the high concentration of water in surrounding. The roots are embedded in the soil, and these cells do not get burst all the time. They have the power to tolerate this hypotonic condition also, although up to a limit, but the limit is so high. So this is the reason behind that is what? It is the presence of the cell wall. Further, we can say, now the next point, if these cells, if they are placed in a hypertonic solution, or if it is placed in a concentrated solution, then what will happen? So normally what is happening? If it is an animal cell, it is placed in a hypertonic, then what happens? The concentration of water is less in surrounding, and therefore the inner cellular water tends to move outside, and the process is known as exoosmosis. So what will happen? Due to exoosmosis, the inner content of water is removed out and the cell tends to shrink. So such shrinking is observed in case of plant cell also. But the phenomena is showing a little bit different trend. When the cells are observed under the microscope, we can see the difference. When these cells, suppose they are placed in the hypertonic solution, the inner water is high. Now you can see this vacuole here. It is so much spongy, it is so turgid. So if from here the water will start moving out. The cell set will start decreasing when it is placed in the hypertonic condition. So water from inside tends to move towards outside. As this happens, the vacuole and another diameter of this inner cellular material will start decreasing. What we mean by this point is that it will start shrinking. You can compare the size of the vacuole first and then the size of the protoplasm. The membrane has started shifting away from the cell wall. So they start shrinking and the process will continue. That means the water keeps on moving outside and finally we will observe that this space inside the cell wall has become empty. And where this material has gone, it has shrunk over there, or the plasma, along with the plasma membrane, it has got shrink over there. Vacuole has no more water here, or it will be vanished. So such type of condition will arise when the cells are placed in the hypertonic solution. What the difference is here is that the cell wall will remain intact. There is no effect over this particular cell wall. The shape will remain definite. It will remain intact. But the inner protoplast, it will get shrink. It will shift to the corner of the cell wall. So this is how we can say shrinking has taken place. Now this process, which is observed in plant cells, in which the cell loses the water when placed in the hypertonic solution. So what happens due to this? The shrinkage or contraction of protoplasm occurs. And this shrinkage or the contraction of protoplasm is termed as plasmolysis. This plasmolysis is a phenomena which is shown by the living cells only. And to prove that, definitely various exercise or various practicals could be done. You can imagine, we can prove this thing. Suppose we have taken the leaves of the plants, and let us say if we have taken the leaf of the plant, and then what we'll do? We will take the section of this leaf and will observe it under the microscope. So what observation will be there? There will be some cells will appear over there with a clear green color chloroplast, and a cell wall would be clearly appeared with the filled protoplast inside. Now next, take these cells. These are placed in the hypertonic solution, and after that again we will observe the cell after some time. So now what we are going to observe, that the outer boundary line was there, but the inner material has got shrunk. Before keeping it in hypertonic solution, let us draw the diagram here. When we have observed it under the microscope, before placing in the hypertonic medium, we will observe something similar structures. This is one of the cell we can see here with nucleus and this thing, but the appearance will be something like this. The cells, they are in close proximity to each other and they are placed something like compartments. This is what we will observe under the microscope. This outer line will be there, then inner membrane will be there, and some nucleus and all this material are going to be observed. Now when these are placed in the hypertonic medium, then what will happen? Later on when we observe it, the appearance will become something like this. This is a single cell we are showing here, and the content is going to get shrink over there. So appearance of this particular section, how it will appear now? Let us assume here now. Suppose this is a part of that leaf. So this outer cell wall will remain like this, and the sections inside this will shrink or move to the side of the cell wall. So this outer boundary is the cell, and the inner membrane along with that content will move towards the side of the cell wall. Further, if we have to see, we can repeat this process in another way. How? We will take the cells, we will take the leaves, we will boil it. Due to boiling, what will happen? The cells will die. Now after this, we will observe it under the microscope. We can observe this kind of structure this way. And if further such cells are kept in the hypertonic solution, then what will happen? They are supposed to be shrinked. But when you observe it under the microscope, no change will appear. There is no such shrinking take place. And this suggests that plasmolysis is strictly a function of a living system. Non-living system will not show the plasmolysis. This is what we want to justify, that although the cell wall itself is a dead membrane, but still it is supporting the life of the cell, and of course it is actually the phenomena related to it is taking place until the cell is alive. Let us study the functions of cell wall now. Now we can highlight the functions with the help of this particular diagram. Cell wall, the first function related to that is turgidity. It makes a cell to appear turgid. It increases the cell set inside. You understand, I hope now, that what we mean by the turgidity. It means the sponginess which has appeared in the cells. So this particular turgidity or that sponginess or that fluffiness in the cell is because of the cell wall. See, when does a cell will appear turgid? It will happen when the amount of water inside the cell is quite higher. So when the cell sap is more. Can you imagine that how it is going to increase? And does it keeps on increasing constantly? No. It is under a controlled manner and it is actually not going to burst away. It is controlled, it appears to be spongy due to the presence of the cell wall. How? That we are definitely going to discuss in detail further. So turgidness is the first important point which is related to the cell wall. Next is, it provides mechanical strength to the cell. What we mean by the strength? It means the power to withhold the various situations, to tolerate the various surrounding conditions. So how is it possible? This mechanical strength is because of the chemical nature of the cell wall. It is made up of what? Cellulose. So this cellulose itself is fibrous in nature and is having the rigid bonds. So this rigidness and this dead nature of these fibers is actually responsible for providing mechanical strength. And you can imagine why this mechanical strength is needed over here in the case of plants. The reason is because they are so stiff. The stiffness is actually because of this type of cell wall only. They are standing, they are exposed to the harsh conditions all the time and they cannot run away from such type of situation. Can you imagine if they are so sophisticated or they are so soft that if the situation is changing, then what will happen? They will fall off. If high speed is coming and they are not strong enough, due to that high speed wind, the plant may fall away. So they have to just tolerate such type of conditions. And so for this purpose, the mechanical strength has to be very high. And therefore the strength is provided by the various chemical molecules which are present inside as well as its dead nature. It helps in exchanging the material. How it helps in exchanging the material? Because it is permeable. So it will support the interaction or the exchange of material in and out of the cell. Besides this, it helps in the interaction between the adjacent cells. How is it possible? This interaction is possible. It is because of the presence of special structures like plasmodesmata and like we can say a middle lamella. So these actually helps in holding the surrounding cells together as well as interacting or sharing the components of the two different cells present inside, and that is possible because of the presence of plasmodesmata. Of course, details we are going to study in the further slides. Now next is protection. This we have studied just now, that how they actually provide the protection to the living cells. One of the example is, suppose if it is placed in the hypertonic solution, then how it makes it to appear turgid, rather it won't allow to get burst over there. Not only this much, it also provides protection in different ways also. So let us study each and every point one by one. So the first point we have said is turgidity. Cell wall is providing turgidity to the cell. What happens actually? Suppose when these cells are present in the hypertonic condition, then what will happen? The water will enter into the vacuole. Because of endoosmosis, the water will enter from surrounding, from outside to the inside. And once it goes in the cytoplasm, it won't remain over there only. It will enter into the vacuole. And this is the reason why the vacuole is so large in the case of these plant cells. So this large vacuole or this vacuole will actually absorb the extra water present in the surrounding cytoplasm. Also, it will serve as the storage site for the various kind of sugars and amino acids, oil, etc. So this water will mix up with that material like those substances, and therefore a sap-like structure is formed. And this sap will actually make it that syrupy consistency of this solution will make it appear turgid over there. So this will get swell up, and this is going to support it will the protoplast inside. It won't allow this membrane to get burst further. Rather, it will be pushed back by the cell wall, and this is why it will appear turgid. Next, mechanical strength. This cell wall, it is so rigid, it is dead in nature. The reason is because it is made up of cellulose fibers. Cellulose, they are forming the microfibrils. This we have already studied. So many cellulose molecules, they are together attached with each other, and they are forming the microfibrils. Then these microfibrils, there are so many microfibrils, and they are interacting or they are bound to each other with the help of other material like pectin and the hemicellulose. So those actually help, they function as a gluing material. So this glue will actually stick these material here. Next thing is that as it is so fibrous and dead in nature, so what will happen? It will provide mechanical strength. This fibrous nature and the dead nature is increasing the mechanical strength of the cell wall. Besides, you must have studied that in certain type of cells, plant cells only, they are having the deposition of other type of material also. Which type of material? See, cellulose itself is secreted by the plant cell, and it starts getting deposited over the outside the plasma membrane. Besides that, in some type of tissues, the cellulose thickening is irregular. So this irregular thickening, it also helps in increasing the thickness and increasing the mechanical strength according to the place where it is present. Besides that, there are the deposition of another compound, complex compound called lignin. Now this lignin is present in several other kinds of tissues, as in case of sclerenchyma. You might have heard about it. Even if not, don't worry, we will study in the later chapter. So this lignin is again a compound which increases the stiffness of the tissue or the cell. So as this stiffness is increasing, therefore it increases the mechanical strength of the cell or the plant. You must have studied that like suppose we take the example of vascular tissue, xylem tissue. These xylem tissue, they are the vascular system, right? They are transporting the water material from the soil to the other parts. Fine. Now as they have to go and function at such a long distance in the plant, they have to be so strong. So that strength is basically provided by these type of depositions in the cells which are present in its surrounding. And therefore we can say the mechanical strength is basically increased because of such depositions present in the cell wall. Further, it helps in the exchange of material. It is possible because of its permeability. They are already permeable in nature, and thus it allows the exchange of various materials across the cell. Now let us assume some more examples to understand it. We just now have quoted the example of xylem tissue. Xylem, which is responsible for transporting the minerals and water from soil to the other parts. Now can you think how is it basically possible? Because the cell wall are having pits in between. These vessels, they are having gaps in their the cell wall. So these gaps actually help in transporting the material in the surrounding cells also. And this is how the permeability as well as these gaps present in the cell wall supports the exchange of material. Next is they can provide the protection to the cell. Now how this protection is provided? We have studied one of the example with the help of that hypertonic condition. So besides that, not only that, they are mechanically strong, they are permeable, they are dead in nature. All these also help the cells or the plants or the organism to withhold the surrounding conditions. For example, the temperature. If it varies, normally what will happen? The plasma membrane may get damaged. The plasma membrane is made up of lipids and protein. When the temperature increases, the flexibility of these lipid molecule increases, as well as protein may get denatured. So they cannot tolerate a very high temperature. But in case of plants, as they are surrounded by the cell wall, then they can provide protection up to a particular limit. So they can tolerate more temperature as compared to the other animals where the cell wall is not present. Similarly, as it is mechanically strong, it is made up of a stiff wall. The cell wall is rigid, stiff or dead. So this actually helps it to tolerate the high wind speed. These are already always exposed to the wind, and the speed of wind is not in anybody's hand. So whenever there is a high speed, the cell wall will help the cell to withhold those particular harsh conditions. Similarly, the atmospheric moisture. If there is more atmospheric moisture, then what will happen? The hypertonic condition will arise, and due to that what will happen? The swelling up of protoplast will increase, and a time will come when it may get burst. But this bursting does not take place, rather it is protected because of the presence of the cell wall. The cell will rather become turgid instead of getting burst. So this is how the cell wall is providing the protection to the cells. Besides this protection, another important function is the interaction between the adjacent cells. Now interaction, how it is possible? It is due to the presence of middle lamella and plasmodesmata. The plasmodesmata you can see in this diagram here. This picture is given. In this cell, the cell wall is having this gap in between. Here also the gap is observed. So this gap will allow the cytoplasm to move out. The material of some of the components of these cytoplasm to get move out. Now further, if it comes out, where it will go? So it is not going to reside in the surrounding condition. Rather, this bridge is continued with the another cell wall of the adjacent cell. Here the another cell is going to lie, which will form the bridge with this thing. We can show you with the help of this diagram. Suppose this is the cell wall of one of the cell, and this is the gap in between. So this gap is going to continue further with the another cell here. So this type of bridge is actually known as plasmodesmata. Here the plasma membrane of another cell will be present. So this is cell number 1, this is cell number 2, and these two cells are interacting with each other with the help of this particular bridge, and this bridge is known as what? It is plasmodesmata. Therefore, they can share the internal components with the another cells also. Further, in between these two cells, there is another material present. They are having a middle layer here. They are sticking with each other with the help of this structure, which is known as middle lamella. This middle lamella, it is made up of calcium or magnesium pactate and is actually helping in interacting the two adjacent cells or holding the two adjacent cells together. You can see in this picture, plasma membrane is here. This is your cell wall, and outside the another membrane or substance is there, which is known as a middle lamella. So this middle lamella will hold the different cells together and will help them to interact with each other. So this is how the cell wall plays such an important role in the case of plant cells or fungal and the bacterial cells. So this is all about this topic here. Next we will study the another organelle or the nucleus or the cytoplasm. The other parts would be studied in the next session. Till then, thank you so much. Have a nice day. Goodbye.